Hello students, welcome back. This is my second video based on the topic that is molecular basis of inheritance. In this topic, we will be talking about the search for the genetic material. Actually, there was a conversation and also we can say like discussion and also competition and there were so much of argument was taken place between the genetic material, whether to consider DNA as a genetic material or RNA as a genetic material. To confirm that only, here we have the some experiments which have been already performed by the so many scientists. In our NCERT textbook, they have given the first one is that is transforming principle. In the year 1928, that is the scientist called as Frederick Griffith have performed an experiment. Actually, he was performing an experiment in the bacteria, call it as a Diplococcus pneumoniae. So Diplococcus pneumoniae is the bacteria. Also we can call it as a pneumococcus, which is also called pneumococcus, also called as a streptococcus. See, this streptococcus and also pneumonia we can also call. See, students, you might be known that Diplococcus pneumoniae means two. Coccus means the spherical shape. Streptococcus means that is shape only. Based on the shape, they two bacteria, same name they have given, and this bacteria name it is called as bacteria, and this is causing the disease called as pneumonia disease. Pneumonia disease is the one which is generally affect the alveolar air sacs, and also our lungs mainly get affected by the bacteria. So the person will suffer from the breathing problems and generally the person will die. So to make one medicine for that and to find out the cure for this disease, that is pneumonia which is causing by the bacteria, Frederick Griffith was already conducting an experiment. When he was conducting an experiment, he has taken these two strains of the bacteria. This we call it as, and one is called it as a smooth strain bacteria. Means the bacteria which is having the smooth in nature, that we call it as an S strain also. And also another bacteria, he also seen that it is showing the rough property, rough nature. So we call it as a rough strain bacteria also, or we can also say it as a R strain also. So two types of bacteria he found, one is smooth strain and one is rough strain. Generally, smooth strain bacteria, that is S strain bacteria, is generally causing the disease, and the rough strain R strain bacteria do not causes the disease. So he wanted to find out how to find out the cure for that. So when he was doing an experiment, he has taken the S strain bacteria, rough strain bacteria, and at the same time he also taken the mice. The first attempt, that is, he has taken the rough strain bacteria. Rough strain bacteria he has taken, that is Streptococcus pneumoniae he has taken, and he injected, that means he has taken that bacterial formation rough strain, he injected into the mice, and later he found that mice was alive, and he confirmed that rough strain bacteria do not have the capability of causing the disease. So he confirmed that this will not cause the disease. Next, he has taken the S strain bacteria, and he introduced into the injected into the mice. Then he found that mice was died. So because smooth strain bacteria causing the disease. Later he found that smooth strain is having a certain properties of causing the disease pneumonia. Later on he heated, that means heat killed the S strain bacteria, means he has given the heat to this, and so that it became the inactive, and so he injected into the mice. So later he found that mice was alive, because when he has given the heat, then what happened, the S strain bacteria became inactive, and also these properties causing the disease, they are just made it as a suspended also we can say. So That's the reason when he introduced into the mice, mice was alive. Later on case, he added heat killed S strain bacteria, and at the same time he also taken the R strain bacteria. So he injected into the mice. Now tell me students, what is going to happen, whether the mice is live or died. So with his surprising, mice was died. What may be the reason for that? So why the mice got died? Because see, S strain is becoming inactive only when it is given the heat to that suspended. So in it, the DNA chemical properties whatever are present, those properties have changed the R strain DNA structures, so that what happened, this strains, this chemical nature have changed the chemical property of the R strain. So R strain now it got the energy, and also now it got the capability of causing the disease, call it as an pneumonia disease. So when he injected into the mice, mice got died, right? So this was the information given by the uh, Frederick Griffith. But what happened? He concluded that R strain bacteria had somehow been transformed by the heat killed uh, S strain bacteria. So, some transforming principle transferred from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become the virulent. So, this must be due to the transfer of the genetic material. However, the biochemical nature of genetic material was not defined from his experiments. So, later on, three other scientists, so those we call it as an Oswald, Avery, and also Mac Leod, and Maclean, Maclean McCarthy in 1933 to 34. So what they have done? So they have taken the genetic material was thought to be a protein. So they worked to determine the biochemical nature of transforming principle in Griffith's experiment. They purified biochemicals of the proteins DNA, RNA, etc. from the heat killed S cells to see which one could transform the live R cells into the S cells. So they discovered that DNA alone from the S bacteria caused the R bacteria to become transformed. How did they perform this? So these three scientists, they have taken the three vessels like structures. So from here they have added, the first one is proteins. Proteins they have added, and they confirmed, they after the adding the proteins, they wanted to see whether they have the causative DNA, or whether they have the same properties. So proteins did not cause. Next, in the second case also, they have added the things, that is RNA. So RNA also, when it is introduced, those properties also did not find. Later, in the third case, they have added the our DNA. So they confirmed that DNA of the S strain has been transformed into the R strain, so that they causes the diseases. So they purified biochemicals, and also they discovered that DNA alone from the S bacteria caused R bacteria to become the transformed. So this is confirmed. So they also discovered that protein digesting enzymes like proteases, and also RNA digesting enzymes like RNases, and also did not affect the transformation. So the transforming substances was not a protein or RNA. Digestion with the DNA did inhibit transformation, suggesting that DNA caused the transformation. So they concluded finally that DNA is the hereditary material, but not all the biologists were convinced. Okay, so why they have not convinced? Because there were no proper information, and also there is a some little bit confusion that still DNA may be evolved from the RNA, and also how this DNA alone can affect this structure. So let's talk in the further videos whether DNA is a genetic material or RNA is a genetic material. Thank you for watching, students. I hope you understood very well. In the further classes, I'll make a best of these videos, students. Okay, please subscribe my channel. Thank you.